All right, as we continue on with chapter number 2, prokaryotic cells. These are our most well-studied types of cells and classes of organisms. They're the simplest because they have no internal compartmentalization. Now, I guess that's kind of a trap because that's not to say that they are simple, but instead it's simply a comparison. They consist of a large and heterogeneous group, so there are members from bacteria and archaea. There are 3 basic shapes. They can be rod-like, spheroidal, or coiled. So examples would be bacilli, cocci, and spirilla would be kind of your classes of shapes. They're typically characterized by their nutritional requirements. So if they're photosynthetic, for instance, what their energy sources are, their chemical composition, and their biochemical capacities. So what they're actually capable of doing from a biochemical standpoint. So we're going to go ahead and start with the description of a prokaryotic cell by looking first at the cell wall. Now the cell wall maintains its shape and basically protects the cell from injury. There is a peptidoglycan layer that is a complex mixture of polypeptides and carbohydrates. Now, in a microbiology course or in lab, one of the things that you do early on is classify a prokaryote based as whether it is a gram-positive or a gram-negative cell. And that has to do with the density and the thickness, I think the thickness more so than the density, but the thickness of the peptidoglycan layer. So a gram-positive bacteria has a very thick peptidoglycan layer, whereas a gram-negative has a very thin peptidoglycan layer. So the next layer within as you peel back the layers of a prokaryotic cell would be your plasma membrane. Now this is a very dynamic layer and dynamic barrier, and it controls molecular transport. It also controls communication with the exterior of the cell. It possesses receptors that are going to detect nutrients as well as toxins, and it's basically going to be it is a literal barrier, but it's also kind of a communicator of what's outside to let the cell let what is going on outside be communicated to the inside of the cell. When it comes to a prokaryote, after you've looked in beyond the plasma membrane, what's within the cell or the cytoplasm? Because keep in mind, you can't really break it down. You don't have compartments within that cytoplasm. Everything's kind of freely moving, and instead everything's just sort of interacting through hydrophobic interactions that are going to aggregate or cause things to aggregate together, or hydrophilic reactions which enable things to basically distribute and disperse throughout. So there are, however, major, I guess you could call them landmarks. One would be the nucleoid. The nucleoid is an irregularly shaped region that contains long circular, keyword there, circular chromosomes. And the reason that I say it's a key word is because I personally have a tendency to think of chromosomes as the human chromosomes, and so I think of human chromosomes as being like this sort of, whereas we're talking about a prokaryote here, in which case you have a circular chromosome. And that circular chromosome, it's significant because what's going to happen with this, long circular object is it's going to wrap in on itself, and so you're going to have these unique coils. And this right here is a little bit closer to what a circular chromosome is going to look like if you were to look at it under an electron microscope or something like that. The nucleoid is host for protein complexes involved in DNA synthesis and regulation of gene expression. So not only is the chromosome circular and it's going to wrap in on itself, but also you've got a bunch of proteins that are associated with that are involved in basically copying that DNA, as well as expressing genes that are found within that DNA. So all these different proteins, which I'm going to go ahead and change color to illustrate those proteins. Bloop, bloop, bloop. Yeah, there's all my proteins right there that are kind of clumped down onto that. Now, the next thing is your ribosome. This is your site of protein synthesis, and it's composed of RNA, ribonucleic acid, and proteins. So, I made a mark on my slide. Now, one of the more, I guess, one of the things that we more commonly hear than not is we hear about probiotics, which probiotics are bacteria that live within our gut. These are also known as our prokaryotic friends. And what these prokaryotic bacteria do is basically help the inner lining of our stomach, or one of the things that they do is they promote an environment where our stomach and our gut tissue is able to do things like absorb different minerals like iron. So they basically make an acidic environment, or one example, like acidophilus bacteria create an acidic environment where iron ions and other minerals are able to be absorbed into our stomach tissue, rather than, if the pH is wrong, then you could have an iron ion get oxidized or reduced, in which case it's not necessarily in the proper ionization state for absorption. So eukaryotic cells, on the other hand, are a bit different because they're more complicated. And they're more complicated, they're more complex, because, well, the most important thing is that they have this membrane-bound organelles within their insides. This complexity allows for a very unique regulation. You can have a process that is regulated at the enzymatic level, but also a process that is not taking place within one organelle within the cell, but it's taking place within another part of the cell, and it's just simply turned off in one part of the cell because of whatever environmental cues. So we're going to continue on with eukaryotic cells in the next video, and I'll see you then.